everybody, we're taking a look at the science behind cattle reproduction and the people that make that possible. We'll be talking to a veterinarian and College of Agriculture researchers and the role that they're playing in advancing cattle reproduction and animal health. Hey everybody, we're here at the Auburn University Beef Unit and we're here with Dr. Jessica Rush. So Dr. Rush, tell us a little bit about your work and how you work with the Beef Unit.
I am Dr. Jessica Rush. I am a food animal theriogenologist at the Auburn University College of Veterinary Medicine, which basically means I'm a reproductive specialist. I work with the Beef Unit as their project vet, meaning I am responsible for all the health care here with the herd.
Well, can you show us a little bit how you work with the herd here?
Yeah, that'd be great.
All right. All right, so Dr. Rush, tell us a little bit about your work in the College of Veterinary Medicine.
So I am an assistant clinical professor, which means I teach students mostly on the clinic floor every day, whether that is in the hospital or in the field. We do everything from herd management to sick cow work, sick goat work with clients to meet their goals and help educate the next generation of veterinary students.
So you obviously work with large animals. How did you kind of come into working with large animals? What drove you to do that?
I grew up on my aunt and uncle's Angus farm, grew up showing calves, absolutely loved it, fell in love with show calves, fell in love with cattle, enjoyed the working outside, being outside, and watching them grow and produce. And so over time, knew I wanted to stay in that situation throughout my life, having that one-on-one interaction with cattle. And then some of my biggest mentors were veterinarians growing up. And my cousin is actually a veterinarian. She's about 10 years older than me. And so watching her, getting to see where her life was leading her, all led me to where I am now.
All right, cool. So how do you work specifically maybe with the Beef Unit? What are you checking when you come out here?
Here we will do pregnancy checks on the mature cow herd and the heifers as well. We do BSEs, breeding soundness exams on the bull every year to make sure that they're fertile before they go into the breeding season, as well as anytime something is having an issue, lameness, illness, or even just not feeling well, I work with their herdsmen to make sure that we're keeping everybody healthy.
Okay. So what does it what does it look like to have a healthy herd?
One, that everyone is in a good body condition. Two, that every cow is producing a calf every year, that they are actually being productive and meeting the needs of the producer.
Okay, cool. So can you show us a little bit of your work? Like when you come into here, what are you looking for?
Yeah, so I mostly just check out the cow, make sure that her health is in good body condition, everything looks good, that she's sound, that she acts like a normal cow. That's what I always tell my students. It's very important to know what normal for an animal is so you can tell abnormal. And so I just make sure that, she holds her head right, her ears are correct, that she stands the way she should, and making sure that she looks like she's feeling the way a cow is supposed to feel and move. If something seems off, we further our exam from there. But almost always we start with what we call a distance exam. So when I look at this cow, when the way she stands, how she stands on her feet, how much coverage she has over her body. She's not thin. She's got a little bit of fat up around her tail head. Her ribs aren't showing, so I can see that she's in a good body condition. The way she holds her ears, I know that sounds kind of funny, but the fact that her ears are in a normal position. They're not up, they're not down. Cattle that feel bad often will let their ears hang a little bit more. That she stands with her back flat. She's not hunched up like something hurts in her abdomen or her chest. They'll often do that standing with their legs underneath them if they're in pain. And just that she's doing the things a cow should do. She's very much looking for some feed right now, but things that I always tell my students, you know, it sounds kind of gross, but cows clean their nose. And so anytime they have a dry nose or a dirty nose, obviously that animal does not feel well enough to be doing that. And so that's a sign that she's not feeling well.
Oh, interesting. So what are some of the common challenges for a herd when you think about health?
Every year is a little bit different nutrition-wise. This year we had a really wet spring and then a pretty dry summer, and so that sets into some different nutritional issues. Nutrition is always going to be the heart of every program, because if animals are not productive themselves, they can't be reproductive, they can't be anything for a herd. And so nutrition is kind of the heart of that, keeping them healthy, keeping them in a good body condition, and that's always a struggle. This year, being through a drought through a good part of the summer, really had a lot of farmers worried about hay for the fall, having enough grass through the summer, and having to supplement hay earlier than expected. So that was a challenge this year for them. And then also making sure the cows get pregnant and stay pregnant. Mm-hmm. That's always what we need to be checking. We always encourage producers to do pregnancy checks early, and if they think they're having a problem, repeat that. But knowing those cows are pregnant, knowing your bulls are good, so that you can have a productive season.
So tell us about, so you do a lot of work on reproduction. That's your main role. So tell us about a little bit more about reproduction. Why is it so important to the management of a farm?
So the whole purpose of having cows is to have calves, to have a product at the end. And so we have to keep them in a good reproductive state to have a product on the end of it, because at the end of the day, the producer is in the business of cattle, and he does have to make a profit for that. And so each cow needs to kind of pay her way every year by having a calf. And so it's so important that they get pregnant at an appropriate time, that they produce a calf every year to pay their own feed bill and for there to be a little bit of a profit moving forward.
Okay, and so then walk us through that. So when you're working with a producer, and maybe they're having, what are some of the common troubles that they may have with cows getting pregnant? And then what does that cycle look like? Do you want them to be pregnant at the same time, or how does that
So the
look?
goal would be to have a breeding season, in that all the animals are getting bred within a specific 2 to 3 month period, so that they're calving within a 2 to 3 month period. And that's for several reasons. One, so you have it much easier for the producer. It's much easier to watch cattle for calving for 2 to 3 months than it is for 12 months. And then also you have a unified product at the end. All of the calves are going to be roughly the same age, the same weight, and it's a more marketable product for the producer to have at the end of it than having calves that are all different ages, all different sizes, that kind of thing.
Okay. So what are some of the common problems that you'll find in a heifer?
So most cattle operations are dealing with things. Nutrition's always going to be an issue. There's usually a lot of lameness that comes up throughout the year. Anything that is stressful or painful can affect fertility in both females and males. And so we always encourage producers to have the bulls checked at least 60 days before going into the breeding season. There's a couple reasons for that. One, with bulls, it takes about 60 days for them to make new sperm. So if they are having an issue that we think they can recover from, we have a chance to check them again before they go in to make sure they have recovered, or the producer has time to find another bull if the bull has gone bad. And then with the cattle herd themselves, making sure that everybody's in good body condition, that they're sound. We can have problems. There are different infectious agents that we worry about that can potentially be issues, different nutritional issues depending on where you are during this in the state, as well as just keeping the animals pregnant. Severe weather, severe changes in anything are all things we have to worry with.
So a producer would essentially, do they call in veterinarians to kind of check? Is that how the veterinarian works with the herd?
Often, yes, they'll call and schedule preg checks, or if they're noticing that a lot of their cows have slipped the calves, that for some reason they're not having as many calves born as they thought there were, we'll come in and do an investigation of the whole herd, looking at the overall health, how many were exposed, to bulls, how many conceived, how many calves hit the ground, and kind of work backwards, looking at their whole herd, the paperwork, what they're vaccinating with, their breeding program, their nutrition program, to see if we can find where there was a hiccup in the process.
Hey everybody, we're here at the Auburn University Beef Teaching Unit on the campus of Auburn University, and we're here with Dr. Paul Dice. So Dr. Dice, tell us a little bit about who you are and what we're going to be seeing today.
Yeah, hi, welcome Megan. So my name is Paul Dice and I'm a researcher and associate professor in the Department of Animal Sciences. And one of my major focuses of my research program is heifer fertility and heifer management. So today we're going to have the opportunity to discuss some of the challenges associated with both properly developing a heifer as well as approaches we're taking in the lab to help producers select those heifers that are going to perform the way they need them to in their first breeding season.
All right, so can we take a look at some of the work that you're doing or how it just all works, comes together?
Absolutely. Let's go have a look.
So tell us, kind of Dr. Dice, how it all begins. What are we looking at and what do you do?
Sure, Megan. So here we have a group of young heifers that represents one of the challenges that producers face when selecting heifers. So we can look at these heifers and see there's some phenotypic differences between them, but nothing real dramatic, nothing real obvious. And the probability of one of these heifers not performing that first breeding season and getting bred easily is pretty high. And so here at this facility, we're able to collect samples from young heifers. The sample we really focus on is blood, and we do that for a number of reasons. One, the blood is a good systemic source of information related to the overall health of an animal. And when we're talking about reproductive fitness, a lot of that is relying on endocrine signals, so signals that end up in the blood that allow the reproductive system to function correctly. And so our reasoning is blood is accessible, so it's going to be something that a producer could collect. And that's really the focus of our research program, is looking at different molecules within the blood to see if there's a difference between heifers that perform the way we need them to and heifers that struggle in that first season. The reason being, when a producer does not get a heifer bred in a timely fashion, it costs them money and it leads to inefficiency in our beef production across the state.
Okay. So what age would these heifers start going into that breeding process?
So heifers are generally bred at least a year of age, usually closer to 14 months old. So it's that period of time between getting weaned, when they're removed from their mother, and prior to being able to be bred, that producers have to sink a lot of costs into to develop them correctly. So our reasoning in our research program is trying to get as far away from that breeding time to be able to give informative information to producers. That would then allow them to make that selection earlier and save those costs associated with developing the heifers. Okay, Megan, so we're here now at the CASIC building where we do further analysis on the samples we collected. So it's important to note the samples we collected were from young heifers, and we need to figure out how they do in their first breeding season before we can really fully analyze the samples. So the first step is going to be processing the blood to allow us to store it long term. Now, as reproductive health has so many factors playing a role, including the environment, nutrition, physiology, endocrinology, we need a very diverse team to properly approach this problem. And so within the lab here, I'm going to introduce Dr. Wellison Deniz and Dr. Priyanka Banerjee, who are key parts of this team. Okay, Megan, now I'll hand you off to Dr. Banerjee and Dr. Deniz.
Awesome. So tell us a little bit about who you are and the work that you're doing.
Thank you, Megan. Yes, I am Dr. Priyanka Banerjee, and I'm an assistant professor in the Department of Animal Science. So I teach principles of biochemistry to the undergrads. My research mostly focuses on biology of reproduction, and so I connect, let's say, all the molecular markers and then connect the immune response and the hormonal signals, the maternal side, and then bring it back to the biology.
Okay.
Yeah, I'm Dr. Wellison Denise. I'm an assistant professor with animal science as well, and I lead the livestock genomics and bioinformatics lab here at Auburn. And I teach animal breeding, an undergrad class, and my research is a bit of looking at what you and Dr. Dice were seeing on the field, like the phenotypes from those animals, and bring a bit of the biology from the research of Dr. Banerjee, and putting together looking at the genomic side. So we look at a massive amount of data, DNA and RNA, trying to integrate it in the way that we can search for those biomarkers and identify which cows have a higher probability of getting pregnant or not at an early life.
Okay, okay. So now we've brought you the samples of blood. So like what happens from this point?
Right, so now because we are trying to get some markers out of the blood since it's the non-invasive process, so now we are going to centrifuge this blood, and then there is a complete protocol that we are going to follow. Centrifuge this blood, extract the nucleic acid, so kind of like DNA, RNA out of the blood, whatever protocols we are following, and then goes in for the further processing. So it goes for or the RNA level, so let's say the protein level or the gene expression level, so we try to see all those levels in the blood.
And then, so once it's done doing all of that, what do you do with the results, or what comes out of that?
So once we have, let's say, the RNA level or the gene expression level, it depends on what level we are going to do it at. If we are just going in to validate some kind of genes, so we will go in for a quick qPCR, and the students are going to demonstrate that. If we are going in more for the bioinformatics side, or if we want it like, okay, which genes are being expressed at a higher level, so then we are going in more for the bioinformatics side.
So you're just kind of like, you're saying, okay, this is what happens. I'm just looking at a certain gene. Right. You're looking at the whole piece of the pie.
Yeah.
Does that make, okay.
Yeah, so what we can think is, we can come from a genetic side to a genomics, and what is going to change is the amount of information that we get at once. If I think about the genetics, I can pick one single gene and analyze that single gene and see how much of that gene is working at that time point. When we say about a genomics level, we are looking at, okay, if we think about the cow, it has 25,000, 30,000 annotated genes. So we look all of them at once and see how they are working. Like, do I have a difference on cows that get pregnant? Do they have genes more expressed or less expressed compared to those that are not getting pregnant? So we have those two groups, one that we classify as fertile, one as subfertile because they didn't get pregnant. What are differences or similarities that they have at the whole genome level? And then we analyze that data and try to find those similarities. But we are more interested in, okay, what is different? What makes this one so good and this one so bad? Although when we are looking at them, they are phenotypically similar. So they look the same, had the same management, but one got pregnant and the other one didn't. So is it genetic? And then we try to identify or pinpoint these markers.
Oh, okay. So Dr. Dice, you've now done all the prep work on the blood, you've gotten it ready, so now what are the next steps?
Yeah, so once the blood is processed and stored, there's some time before we actually find out whether those heifers perform well or poorly in their first breeding season. Once we get that information is when we can really start asking questions about their physiology and potential molecular markers. So we usually start by comparing their physiology by looking at their uterine tracts and ensuring that there's nothing obviously different between our good and our bad heifers. This is an important step in the process because if there is a way physically that we can detect reproductive problems, that's the least invasive way we can answer that question, and the most cost-effective way we can answer that question. So we usually start with the physiology, looking at the uterine tracts and ensuring that the heifers were mature and were capable of breeding. What we often find, though, is those heifers that perform exactly as producers want and those heifers that do not perform well physiologically are not distinctive, they're identical. And so that's where we really rely heavily on molecular answers to these questions.
Okay. So now we're kind of diving deeper into the molecular level. So walk us through what happens in this lab.
Yeah, absolutely. So for the molecular side, like we are looking at the gene expression. So we have extracted the RNA that you just saw in the protocols. Now we have to see the gene expression levels. And in order to check the quality, we have this instrument of Agilent bioanalyzer. So we load our RNA samples in this chip, and this chip goes into this instrument to see the RNA peaks, like if it is contaminated, if it's degraded, or how are those peaks. So this is the PCR workstation, and what we do here is to check the levels of the RNA as well. So there is a complete protocol for that we convert the RNA into a cDNA, and then we check the levels, come and check over here. So in order to do that, we first have to dilute the samples and then make it into a plate for all those that have the better RNA quality. So all those that we take in, make into a plate, and then it goes into a qPCR machine. So here we have the gel dock. So actually what we do, we place the agarose gel, which is with the PCR products that we just amplified, and we place the gel over here. And then what we do, we actually visualize them on the screen. So what we have the ladder over here and all the genes and the specific products. So if we have, like, each one of them is one specific size, and each one of them depicts like what size it is amplifying into. So in this way, we will be able to know that the gene we are looking for, it's actually getting amplified or not, or it's nothing. And we are able to see, like, all right, in the fertile and in the subfertile heifer, how are these genes acting? Are they getting amplified in one group or in both the groups? So what is the difference of the amplification in a good heifer and in a bad heifer? Whether the gene that we are looking for is there, it's amplified in the good one, or it's not amplified in the bad one, or the other way around. And now in order to see a global picture on how all of these genes are acting and how these targets are behaving, how is the expression, we go in more for the sequencing projects that we run in this lab, and then we also go in for more of the bioinformatics analysis.
So Dr. Dinez, so now we're kind of looking more at the gene, how the gene has been expressed, correct? And like
Yes.
You're taking a really deep dive into that. So what's that look like for you and the work that you're doing?
Yeah, like we can look both from the RNA side or the DNA side. Like the questions are going to be different. When we say about the RNA, so one point is like with Dr. Banerjee was doing, you can look one gene at a time. Here, when we say about the bioinformatics side and the genomic side, we want to look at the whole genome at once and kind of taking a picture of that moment, what is happening in the physiology of that animal. So basically what we do is like, okay, we collect those samples, do that prep that we are doing in the lab, wait for the pregnancy results, and then we start like, if we want to see the expression profiles, our main question is, what are the difference between the expression profile of a heifer that got pregnant and one that did not, although we managed them in the same way. So our main point here now is isolate that RNA, see the expression, and then we try using some bioinformatics and computer tools, some machine learning models. Our idea is to do those comparisons and identify specific genes that are different, and what would be the biological process that are related. Like, is it an immune process, is it a hormonal process that are imbalanced between those two groups, and what are the genes that are related to those processes that are leading to those differences.
So you obviously work with a lot of vet students. You know, how are you training them, and maybe like how are you like bridging that gap with students that maybe haven't worked with large animals and don't know what it's like to be on the farm?
The ones that haven't been on the farm, the biggest thing is trying to make them feel comfortable and submerging them in it as fast as we can, because it's a very short period of time that they have with me directly on clinics throughout their career. So Often it's easier to assign those students to calves or small ruminants or camelids to get their feet wet just a little bit to where they feel comfortable. And then we have to run through how to move cattle, how to be around cattle so that they feel safe while doing it. That's usually their biggest challenge, is that they are scared of the animal and they don't know where to be and where to stand. And so by putting the ball in their court, putting their hands into it automatically, they tend to do a whole lot better than just talking them through it, actually getting them in there and getting ready. And then making them take ownership of their cases. That's always my goal. I want them to make the decisions. We can always sit down and talk about why we need to revise our plan, but they need to be the doctor, make the plan, make the decisions. And then we come back, sit down, and go over why something might need to change, whether it's an economic reason or a better plan, better idea, new thing they haven't thought about or been exposed to. Every opportunity is learning, and that's what we want to do, but we want to create that problem-solving situation in their mind, because large animal is a lot of adapt and overcome.
Yeah. Is there any other advice you'd give to students now as they're working to either enter vet school or just enter the industry?
Mostly just take every opportunity you can and enjoy every minute of it. Every interaction I had led me somewhere, and you can learn something from everyone you meet, and so it allows you to take each step forward. Not every step may be exactly where you want to go, but they'll get you where you want to go if you hold on to your dreams and what you want to do. The right pathway will find you.
Well, thank you for showing us a little
Thank
bit
you.
and talking to us about what the work that you're doing. We appreciate all that you do just for the university as a whole, working with students and just empowering them to be better veterinarians. So each of you have a lot of undergraduate students in your lab. What are your students doing in your lab, and maybe what's the value of having them in the lab?
Yeah, so I think in all cases our research program has provided a good opportunity for undergraduates to experience primary research. And it's generally, I always suggest undergraduates try to test the waters before they go all in on a graduate program. So it provides a really good opportunity for them to pick up skills, learn the scientific method, and make an informed decision before they sign up to a multi-year graduate degree.
Mm-hmm. Yeah, the labs that we work with is like mostly student driven, mm-hmm. and so they have a good opportunity to learn most of the skill, and we are also happy to train the next generation scientists so they can go in and whatever they have a great experience of whatever is going on the field to bring it back to the lab, understand it more, and then apply it back to the field. So I think it's much more specific for them.
So the best way for them to really get involved is really just to ask, hey, I'm interested, or I want to see what you do. Mm-hmm. What do you have? And you'll usually help get them connected some way.
Absolutely. In all our programs, we have a very open door policy. Even if they just want to see a little bit of what's going on and make a decision from there, how vested they want to become in our research program.
So what are the characteristics that make an undergraduate researcher successful, or even a graduate researcher successful in your lab?
For me, it's curiosity and enthusiasm. If they're interested in getting the answer, I can mentor them a whole lot easier than if they don't care about the answer. They're not going to be in Saturday morning, late at night, when we need them to be in. So for me, I really just look for that spark of curiosity and the enthusiasm to get where they need to go.
As an undergrad, thinking long term, and sometimes I talk with my students in the sense, okay, you don't want to be an animal breeder, but what are the knowledge or skills that you need that can help you to be successful in any other path that you decided to take? So we try to see in this way as well, like skills that you can use in multiple opportunities or in multiple areas depending on where you want to go. If you want to go to academia, you have that research method, but if you want to go to industry, what were those skills that you can translate and use on your work on a daily basis?
That's really great advice. And I just want to thank y'all for letting us tag along with you today and showing us the work that you're doing and the impact that you're going to be making on producers and how they're managing their
Thank
herd.
you all. It was wonderful, and it was a great experience for all of us as well.